Okay, to introduce this session, I'm Doug Lauffenburger. I'm head of the Biological Engineering Department here. And quite honestly, I think my main contribution to this symposium happened 15 some years ago, because when we started the new department, the very first senior faculty person we hired was Dane Wittrup from University of Illinois. Very first junior faculty member we hired was Daryl Irvin from Mark Davis's lab. So we put a big bet on immunoengineering a decade and a half ago, and I think it's paid off and grown splendidly, and today's symposium is a nice manifestation of it. So for my own contribution then to start this session, Dane mentioned that our lab is interested in the complexity of biology and how to think about things, integrating multiple threads, pulling together how networks interact of molecules and cells. So that's what this will be. And so this first subtitle here, it will be methodology, and that's a big part of it. And then the second part is sort of the take-home principles of things that we're learning. So methods of doing in vivo multivariate analysis, and then some things we've learned about the communication going on in these kind of environments. I'd like to first start with the team here, Kevin Haggis at MGH in Beth Israel, David Burrell at Boston Children's on a lot of the in vivo work, and then postdocs Ken Lau, Jesse Lyons, grad student Sarah Schreier, and funds from NIH and the Army. So here's the key objective of our lab in a lot of ways these days, is we'd like to understand how to intervene in predictive matter in complex diseases, and especially immune system associated. So in the previous session and in ensuing, you'll see all the power of ways to intervene with small molecule inhibitors, with antibodies, with cytokines, with cells. There's many ways we can intervene in the immune system in this disease. And the question is, given the complexity of all the interactions, how do we understand how any intervention, any perturbation will find its way through all these threads to give the ultimate tapestry of outcome? So what are the ways to look at it? One place that there's a lot of attention these days is to take individual immune cell types and kind of one by one interrogate them and say, what do these cells do and what does this cell do? The problem with that, of course, is it should be appreciated that even if you have the same cell type, if you put two of them together, they'll do something different than if you have only one. And then as soon as you introduce another cell type, they'll do something different. So the positive and negative synergies in terms of cytokines produced, genes expressed, just by putting multiple cells together, either of the same type or multiple types, make it very difficult to add up in a linear way what individual cell information gives you. So then you say, well, then what we really got to go to is the communication network among different immune cell types, and you can find lots of data in the literature and illustrative depictions like that. But the question that has nagged me, coming more from originally a cancer biology field, is what about the rest of the tissue cells that the immune cells are residing in? So in particular, if you're interested in a tumor microenvironment, or in this case, intestinal inflammation, other types of diseases, the immune cells are talking to each other, but they're also being interacted with by stromal cells, epithelial cells, and so forth. And in fact, you can find in the literature some nice depictions of not only the crosstalk of communication of cells among the immune system, depicted by hematopoietic here talking to themselves, but also with epithelial cells, mesenchymal cells, nervous system, endothelial, and so forth. So this really is the cell-cell communication network going on when you're talking about what an immune system is doing in a tissue. So how do we begin to interrogate that? Well, from an engineering background, what one realizes you can't really start with theory that's going to predict everything. One starts with experiment and measurements. So you really have to interrogate the system experimentally with molecular and cellular measurements across multiple components, molecule types, cell types, associated phenotypic measurements, cellular tissue, in order to figure out what's happening at the outcome level along with the underlying mechanism level. Need to do this across a landscape of diverse conditions. Any measurements that you make under one set of conditions, you don't know what's epiphenomena and what's really crucial. And then finally, it may not be big data for whatever name that is depicting, but generally you need computational analysis then to pull out hypotheses and insights and predictions out of these. So that's really what we're after. We're going to start to unravel some of these threads. What I'll talk about today is in mouse models, although we've been doing this in human subjects lately as well. So it has to do with perturbations and treatments, maybe under different genetic backgrounds, making measurements from in vivo, and then phenotypic outcomes, and seeing if we can, as I said, start to unravel some of these threads. So I'm going to give you two examples. They're both related. But they're not meant to be a disease model. I want to emphasize that. These are model approaches to see, does our methodology work? Okay, proof of concept. Can we learn things? And then when one develops things that people really believe as disease models, whether they're mouse or human, then we really know what to do. So the very first one is just a very simple acute intestinal inflammation in mouse models, just TNF induced. So mice of various genetic backgrounds will be injected with TNF at different doses. And the TNF is going to do something. It's an inflammatory stimulus. And in particular, what we're interested in is what it does to the epithelium as a barrier function. And so I'll talk about measurements in a minute, but we'll talk a lot about the measurements of hyperproliferation and apoptosis. And the TNF is working on a background of the immune cells, so it's now perturbing the immune cell network and the way the immune cell network is interacting with the epithelial cells. So just to run through what some of the measurements look like. So under this cycle of genetic backgrounds, treatments, taking tissue at different time points, first thing measuring might be epithelial cell proliferation and apoptotic death. That's our surrogate for pathophysiology here. And you can see it by immunohistochemistry, what's happening with proliferation or apoptosis. You can quantify it by taking the tissue samples and see things like there's a dose response and time response. So higher doses of TNF give you an earlier cell death. There's perturbation of the immune system effects. So if you do this in RAG1 knockouts, you both get a highly aggravated cell death and much earlier. So there's something protective about what the immune cells are doing in this particular model. So to try to sort all that out, here's phosphosignals from within the epithelial cells, measuring across the number of the different kinase pathways under the different TNF dose conditions and knockout conditions. You see lots of changes. Just about all the pathways are changing. Measurement of cytokines and chemokines, about 50. This is just a subset depicted here. Again, dose response effects of TNF and the knockout effects. And just about every cytokine and chemokine is affected, although very modestly. So, okay, now I'm changing my signaling pathways inside the epithelium. I'm changing my apoptosis and my hyperproliferation. They're seeing different cytokine milieux. Now, by flow cytometry, measuring cell types, again, under the different conditions. What's changing with the number of NK cells or Tregs or CD8s, CD4s, B cells, different types of macrophages, neutrophils, plesiocyte dendritic cells. And again, everything's changing modestly. So what you see is modest changes at the cell types, modest changes at the cytokine levels, modest changes at the signaling level, but yet drastic things are happening in terms of pathophysiology. All right, so to sort this out, one brings some computational analysis. Doesn't have to be sophisticated. We've learned a lot of things from some very simple multivariate dimensionality reduction, matrix analysis. In this case, something called discriminant partial least squares regression. And I'm just going to take you through some parts of the story just to see what one can do. So this is about a couple dozen of the cytokines and chemokines and how they change with time after the TNF stimulation under different doses and whether you've got the wild type or RAG1 knockouts. And again, you see changes in cytokines and chemokines being produced under the various conditions. Now, what's actually associated with the apoptotic phenotype? Well, what this analysis allows you to do is to essentially cluster or aggregate either the most modest effect, the low-dose TNF, very late and low apoptosis, higher dose earlier but low, and then with the RAG1 knockouts, early and high. And now you find, from what's called a loadings plot, how these different signals are in a co-correlated way, in a multivariate way, associated with these various phenotypic outcomes. It's very interesting that a very early level of MCP1 or CCL2 is strongly associated with protection against apoptosis in the epithelium. And I want to point out that is this difference between this blue and this red. Okay, so out of all this sea of changes of blues and reds and so forth, this is pulled up by the computation as the most decisively protective against the apoptosis induced by taking out your lymphocytes. I don't think you would have just stared at this picture and guessed that. Well, so you want to test it, and in fact it happens. So you can deplete MCP1 with an antibody and put the resulting changes in the cytokines in the model, and it says yes. It should shift it from, even if I had wild type, where I don't have much apoptosis, low dose, but if now I deplete the MCP1, it shifts up that it ought to look like I've knocked out the lymphocytes entirely. And in fact, if you actually measure the apoptosis, that's exactly what happens. That here's the wild type mice, low dose TNF, just deplete the MCP1, and now you get the same kind of early and aggravated apoptosis as if you had knocked out the lymphocytes. So you can, this computational analysis can pull out threads, and then of course you can go back in and test them in dedicated manner. Now you might ask, okay, where's the MCP1 coming from? Is it coming from the lymphocytes? Because when we take it out, things change. Well, no. For folks in the field, it's typically considered to be produced by immune cells as a macrophage chemoattractant. Turns out in this context, it's neither of those. It's mainly made by the epithelial goblet cells, and you can see this by taking out and looking at message at the various cell types. And it really doesn't do anything to change the macrophage levels. And we've dug into these things. So neither of the classical things is what's operative in this particular model. So it's made by epithelial cells, not by immune cells, and it really doesn't affect macrophages. So what is actually governing this? Well, it has to do with epithelial cell differentiation into goblet cells. Another way to perturb that is by a Notch inhibitor, because goblet cell differentiation from intestinal stem cells is repressed by Notch signaling, so we have an independent tuning of what the epithelial cells are predicted to be doing in this model. So it turns out if you do that with a gamma secretase inhibitor, yes, that you can change the amount of MCP1 by increasing the amount of goblet cells. So just by tuning up the goblet cells, you get more MCP1, and in fact, then you get less apoptosis. So now you go to the cell types. So the lymphocytes are doing something to the goblet cells. The goblet cells are making MCP1. What's that doing? It's not really changing the macrophages. Well, so now you go back and look at the flow cytometry, and what you find, in fact, that the biggest change induced by either the RAG1 knockouts or depleting the MCP1 is in the plasmacytoid dendritic cells. So that's predicted to be, that's now the intermediate from lymphocytes to goblet cells to something to epithelial cell apoptosis. And now you can test that again by depleting various cell types. And now, as predicted by this analysis, depleting the plasmacytoid dendritic cells makes, from wild type mice or from RAG1 mice, now makes them behave like wild type. So if I have RAG1 knockout mice that I have an aggravated apoptosis, now if I deplete the plasmacytoid dendritic cells, they're like wild type. They're protected from apoptosis. Okay, and now why is that? Well, now you go back in and you've done the same measurements on cytokines and so forth under all these perturbations. So we build an even stronger model. And here's our MCP1 over there that was associated with protection. And now under these conditions, what comes out is interferon gamma is strongly associated then with the aggravated apoptosis that you get with the plasmacytoid dendritic cells. And you can test that here under the different conditions, add interferon gamma for wild type. yes, you get the increased apoptosis of the epithelial cells, take the RAG1s, and deplete interferon gamma, and it goes back toward wild type. So the point is that, and this is just one partial thread. There's many other threads going on here. I'm not claiming this is the decisive thread. This is a thread, and we could go back into that same data set and perturbations and find many, many other threads, all of which come together to create the integrated model. But the point here is that adding TNF, what really determines the strength of its action on the epithelial cells is this crosstalk between the lymphocytes that are doing something to induce the goblet cells to make MCP1. What that's actually doing in this context is blocking plasmacytoid dendritic cells from coming in, because when the T cells aren't there and the goblet cells don't make the MCP1, then they do come in and they induce interferon gamma. And by signaling pathway models that I don't have time to show you here, it's actually the synergy between interferon gamma and TNF alpha that causes the crashing and burning of the epithelium. So the point here is, by studying the immune cells by themselves, we never would have seen this thread. It's the back and forth between immune cells, tissue cells, immune cells, tissue cells. That's the thread you've got to follow all the way through. So let's just push this a little bit farther into a chronic model that was an acute model. And this is just a very ordinary T cell transfer model into the reg knockout mice. And again, not meant to be an IBD model, something like that, but again, a proof of concept. So you can put back in naive T cells, and depending on the individual, and this is really good for this kind of systems analysis, variation among individuals is good, because these are statistical models. The more variation you get, the better you can sort out signal from noise. If every mouse behaved the same way under the same treatment condition, you've wasted all the mice but one, okay, in terms of the way this works. Some other individuals, you get much more severe inflammation. If you transfer Tregs, you essentially get no inflammation. And this is measured by the crypt height, the hyperproliferation in the intestinal epithelium, and by infiltrate. We can measure the infiltrate of T cells, macrophages, neutrophils, plasmacytoid dendritic cells, along with making the same kinds of phosphoprotein signaling measurements, cytokine measurements, cell types, along with mucosal thickness. So here's now our phenotype under these conditions. The same kind of multivariate matrix dimensionality reduction modeling says that, okay, we can really separate the individuals that get very severe inflammation from the naive T transfer, individuals that get a much milder, and then the ones that get essentially none because of the Tregs. And now we ask, what are the molecular mechanisms associated with that? So again, looking at the loadings plots, cytokines and chemokines, some are very strongly associated with severe, some are very negatively associated with severe. Like previously, remember MCP1 was way over there for the TNF alpha. Here, if we're looking on this side, there's a number of chemokines that are very strongly associated with the severe inflammatory pathology. If we go ahead and look in the signaling pathways, and again, we can demonstrate that this is largely in the epithelial cells, not in the immune cells. Very strong activation of the PI3 kinase mTOR pathway is associated with the severe inflammation. GSK3 looks red, downregulated, but in fact, since it's an inhibitor in this pathway, it's an activator of the pathway. So that would say that if this is true, if this signaling pathway is implicated, then something like a rapamycin, mTOR inhibitor should have influence. And it does. So first of all, you treat the animals with rapamycin. Just measure the cytokines or the signals again. Put them back into the same model, and before you even measure the hyperproliferation, you say, what does it predict? And it predicts from what is done to the cytokines and chemokines that it should shift you to a milder inflammation from the more severe to the left. You measure the mucosal thickness, and that is what happens. So again, the model can predict based on the measurements of what rapamycin does to the cytokine microenvironment. And again, I want to emphasize, in the IBD field, mTOR has been around a long time. in most quarters that I've read, mainly presumed to be operating in the immune cells. Again, in this context, it seems to be mainly living in the epithelial cells. The epithelial cells is where the action is, again, for modulating the cytokines and chemokines. And in fact, if you do a time course, that seems to be the case, that if you have an mTOR inhibitor, the very first thing that happens is changes in the epithelial cells, essentially the proliferative to differentiated fraction. So the first thing that happens is what rapamycin does, the mTOR inhibition, is changing the epithelial cell differentiation state. Then you change the cytokines, and then 2 days later, you change the immune cell repertoire that's coming in. So temporally, you treat with this inhibitor, first thing you do is change the epithelial cell state, then that changes the cytokines, then that changes the immune cells. So the picture is something like this, that I've got this complex milieu of microbiome and immune cells and the differentiating intestinal epithelial cells from stem cells to goblet cells and enterocytes. And in terms of time courses, when you're perturbing it, whatever your perturbation is, at least it goes through the epithelium to the cytokine microenvironment, which then modulates the immune cells and, of course, continues to feed back. And so turns out you say, OK, can we actually test this in the same way? You can use the Notch inhibitor to just change your epithelial cell state independent of the mTOR inhibitor and find the same things, the same shifts in chemokines, the same shifts then in ensuing immune cell infiltration. Okay, just by changing the epithelial cell differentiation state. And then another test of this is, OK, can we take this out in vitro and with David Brohl at Children's Hospital to grow intestinal organoids. And there's different ways to manipulate these intestinal organoids with different treatments to make them more stem cell-like, more enterocyte-like, more goblet cell-like. So you can actually put these organoids in different states. And now if you measure, under these conditions, what sets of cytokines and chemokines they're making, put those back into the model that we got originally from the very first set of T-cell transfer. It says, what is the state of the cytokines? The ones in the treatments that are undifferentiated, that set of cytokines is indeed the set that's the same as if I got the severe inflammation from the actual T-cell transfer itself. If in the organoids, we put them under conditions that are more highly differentiated and now measure what cytokines and chemokines they're making, put them back into the in vivo model, it says these should behave in a very mild way, almost as if the Tregs, no inflammation. So the cytokine chemokine signature you get from the intestinal epithelial organoids, depending on how you treat them, is actually predictive of the state of what would happen from the T-cell transfer, which, as we've argued, largely modulates the epithelium. So I'll end here, and I think we're good on time. So if our objective is to understand what would happen if you modulate cell types with cell type depletions or additions or programming or add antibodies or add cytokines or deplete them, our point is that what happens with the resulting pathology or effects of treatment isn't as simple as some direct line of that perturbation to your pathology. There's a lot of threads that it runs through, and a big part of that thread isn't just the communication between the immune cells themselves, but it's the communication between the immune cells and epithelial or mesenchymal cells and back, and that's the communication network that needs to be looked into as well. So with that, I'll just make sure you see that this is largely due to my collaborators Kevin Haggis and David Brohl over at Harvard Med School, postdocs Ken Lau and Jesse Lyons, and graduate student Sarah Schreier, and funding from NIH and the Army. So thank you for your attention.